Hello friends, welcome back to my channel, and today in this video we are going to study about how can we map the bacterial genes with interrupted conjugation. In the last video we learned about conjugation, and today we'll see how we can use this method to map any bacterial genes. Before that, few points that we already learned in the last video, just revising it. That is, one cell having F plasmid can transfer this to another cell. Correct? Whenever the transfer has been taking place, the site called ority. From here, the chromosome will be nicked and the transfer will take place. So the transfer will take place. This blue section will enter first, and after that remaining chromosome will enter into the recipient cell. So that means we can say that this transfer of genetic material, it has a direction, or we can say it has a polarity. We can use the word polarity also, we can use the word direction also. In a particular direction only the gene will get transferred. If suppose in the chromosome the genes are present like B, then D, then A, and then C. So when it will getting transferred into the recipient cell, because the blue section will enter first following by the rest of the chromosome. So when this particular chromosome will enter into the recipient cell, the first gene that will get inserted into the recipient cell will be the B gene, because B gene is present just after this ority segment. So the B gene will get inside. Then the next gene that will enter into the recipient cell will be the D, then it will be the A, and then it will be the C gene. Clear? Now one more thing here is, suppose this is the chromosome and from here it is getting inserted into the recipient cell. Now suppose the B gene is present here and D gene is present here, whereas A is present here, then C is present here. So here you can see it will take 100 minutes for the entire chromosome to get transferred from one cell to another cell. So 100 minutes it takes for the entire chromosome to get transferred. Suppose, for example, if we interrupt or if we stop the process of conjugation at 20 minutes, for example, I'm telling you. So maybe after 20 minutes in the recipient cell, we can only find gene B and D. After halting it for 2 minutes maybe, then also here suppose 2 minutes. After 2 minutes if we stop the process of conjugation, still we are getting B gene and D gene. Maybe after 35 minutes if we stop the process of conjugation, we'll get B gene, D gene and A gene. So that's how we can interrupt the process of conjugation at different time intervals to decide the position of genes present on that particular chromosome. Now for this, we always take an HFR cell and F minus cell. Why not F plus and F minus cell? Because in F plus cell, the plasmid will get only transferred to F minus cell. Whereas if we say it is an HFR cell, that means this plasmid is actually integrated into the chromosome. So from HFR to F minus cell, that means the chromosome is actually getting transferred, which takes approximately 100 minutes for the entire chromosome to get transferred. So we can halt the conjugation at different time intervals to know the distance between two genes on a particular chromosome. Now let's solve one question. So the thing is, an HFR cell, which is positive for all these genes, galactose, histidine, biotin, purine, and glycine, is mixed with F minus strain, which is negative for all these. But the strain consists of streptomycin resistant gene. When two such cells are mixed and then plated in a medium containing streptomycin, then what will happen? We can select out the cell that received the gene from HFR cells. Now nothing else we have to read. Only we have to read is what? What is the question? The basis on these data, give the order of the genes and indicate the minimum distance between them. We have to give the order of the gene also and the distance between the genes also. Here, when we map the gene on the basis of conjugation, the gene distance is given in minutes, not in map units. Correct? Here, we indicate the gene distance on the basis of minutes. Now, as you can see, glycine. This is a chromosome. Glycine, when we halt the process of conjugation at 3 minutes, the gene obtained was glycine. So definitely glycine will be at the first position. Then histidine, 14 minutes. Then biotin, 35 minutes. Then galactose, 36 minutes. Then purine, 38 minutes. So here it is 3 minutes, 14 minutes, 35 minutes, after 36 minutes and after 38 minutes, this gene has been obtained. Now what is the distance between these genes? 11 minutes, 21 minutes, 1 minute, 2 minutes. So this is the order of the gene and this is the distance between the genes. Now with the help of this information, we have to decide the order of the gene and also the location of F factor. Now one point to note here. I already told wherever the F factor will located, from here only the gene will transfer to the recipient cell. So in case of gene order, wherever we'll find this F factor, after that only the genes will be present. So the first thing that gets entered into the recipient cell is the F factor, and the genes present in a sequence behind the F factor will enter into the recipient cell simultaneously. Clear? Now, this is a circular DNA. How I know? It is written here. Now, if we see the HFR5, the sequence is M, Q, P, N, R, O. The gene that entered first in the recipient cell is M, then Q, then P. So what will be the location of F factor and its polarity? For HFR5, definitely the F factor is located here. This is for HFR5. That's why the F factor entered first and after that M, Q, P, N, R and O. Done. HFR4. It is the first gene that entered is N. N, then R, then O, M, Q and P. So for HFR4, this is the location and polarity of F factor. From here it entered, that's why the first gene that entered into the recipient is N, then R, then O, then M. HFR1, O, then M, then Q, P, N and R, O. So here, for HFR1, the location of the F factor and the polarity is somewhere here. HFR9, which gene entered first? Q. After Q, M. So it is moving in the reverse direction, as you can see. So Q entered first, then M, then O, then R, then N, then P. So here will be the F factor. But the orientation is opposite compared to rest all other HFR F factor. Here the orientation is anti-clockwise with HFR9. Clear? That's how we map the genes and determine the position of F factor, its polarity or the distance between the genes. Now this question is for you. You have to construct a genetic map and let me know the distance between the genes. So if you found the video informative, please do like, share and subscribe the channel and hit the bell icon for the next video notification. Till then, stay safe, stay happy.